KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing the location to the process of photosynthesis. Now we are going to discuss. Location means what is a cell organelle or what is the name of the cell organelle which is responsible to the process of photosynthesis. So there the deep information we will discuss in this video. Now let us start to discuss. Now the mesophyll cells in the leaves have large number of chloroplasts. Here first point here, if you see mesophyll cells means, did you remember or not? While discussing about the anatomy of flowering plants, so we have discussed in a dicot plants, palisade parenchyma and sponge parenchyma likewise. So mesophyll tissue is divided into two types we see, isn't it? Means just below the upper epidermis we have seen the presence of palisade parenchyma, and below the lower epidermis we have seen the presence of sponge parenchyma. Likewise in a monocotyledons, so just mesophyll tissue only is present. It is not differentiated into palisade and sponge. Means mesophyll tissue is not differentiated into two types in monocots, and in dicots mesophyll tissue divided into two types, palisade and sponge respectively. Already we have seen. Now mesophyll cells, so these mesophyll cells or the mesophyll tissue have large number of chloroplasts. So means here the chloroplast is a cell organelle or is a kind of cell organelle, helps to the process of photosynthesis. Okay, now usually the chloroplasts align themselves along with the walls of the mesophyll cells. Okay, so one more time, usually the chloroplasts align themselves along the walls of mesophyll cells, so such that they get optimum quantity of incident light. Sir, why sir here the chloroplast cells so arranged or very near by the walls of the mesophyll cells means, so here, so and here the arrangement also they will do themselves, means here the chloroplasts arrange or align themselves where along with the walls of the mesophyll cells. So when these chloroplasts are arranged along with the walls of the mesophyll cells now, so they may get sufficient quantity of the or quantities of the incident light. Suppose if incident light if they get more means, so the process of photosynthesis and the productivity will be more. So that's why here the chloroplast cells or the chloroplast cell organelles are arranged at the walls of the or nearby walls of the mesophyll cells. Now within the chloroplast there is a membranous system, so consists of is the important bit. So within the chloroplast there is a membranous system. Membranous system consists of grana, stroma lamellae and matrix stroma. Okay, means here grana, stroma lamellae and stroma. So these three are membranous system we can consider. Okay, now there is a clear division of labor within the chloroplast. Means here the each and every compartment that has been divided very clearly, that we can study furtherly. Now the membranous system is responsible for the trapping of light energy. So means here if you see the diagram, here the diagram also is visible, I hope. Now here if you see the structure, here the sac-like structures are there. So many means bag-like structures are there. So now here the tubular and the sac-like structures, when all these are all comes together and form a grana. Actually if you take one sac-like structure or simple sac-like structure or the bag-like structure, no, tubular structure, so that one each and every structure we are calling as thylakoid. Now the group of thylakoids comes together and form a grana. So sir, why sir, what is the reason why these thylakoids comes together and arranging as a like a stalk means, or the stalk of thylakoids means to absorb the sufficient quantity of the sunlight or optimum quantities of the sunlight. So that's why here what is the point now we are learning? The membranous system responsible for the trapping of light energy and also for the synthesis of ATP and NADPH. So thereby here we are getting one important point, that is so by the process of light energy, so definitely in the thylakoid membrane or on the thylakoid membrane, so when the thylakoid membrane receives the light energy, so thereby which one is going to be synthesized? ATP as well as NADPH. So ATP as well as NADPH are going to be synthesized in the light reaction. This you don't forget. இப்போ இன்சிடென்ட் லைட் இருக்குமா, இன்சிடென்ட் லைட்னா ஜஸ்ட் so what is a light which is coming from the sunlight, you can take like that. So now it will fall on the leaf surface, so thereby so here what is the thing, when the leaf when the light is absorbed by the leaf, what is happening, starts to synthesize as the ATP as well as NADPH in the light reactions. So thereby here somewhat light energy is going to be synthesized in the form of ATP as well as NADPH. So means here sir, where the particular location to absorb the light means the particular location that we can be seen in on the thylakoid membrane. Means here the thylakoid membrane, why sir it has the capability means, so on the thylakoid membrane we will notice the presence of pigments. So we will notice the presence of pigments. So means, so because of the presence of pigments here on this one, so what are the pigments are present on the membrane? No, starts to absorb the sunlight, so thereby gradually involves in the preparation of light energy in the form of ATP as well as NADPH. Okay, next one in stroma. So first of all if you see the diagram, suppose if you see the diagram here, இப்போ பார்த்தோமே இங்கே the grana is nothing but இப்போ thylakoids எல்லாமே thylakoids ஒரு பல்க் மாதிரி ஒரு குரூப் இருக்குதுன்னா, ஸோ ஒரு செட் பை செட்டப் இதுதான் grana. So likewise here so many number of granas are present, means approximately 20 to 40. So we will notice. Okay, so here now if you see there the grana, the group of thylakoids. Now here what is the use of this grana means, absorbs the sunlight. So because of the absorption of sunlight by the thylakoid membrane, no, so starts to prepare the ATP or starts to synthesize the ATP as well as NADPH. Okay, அப்புறம் பார்த்தோம். So empty space is there, no, this particular empty space only we will be taking as a stroma. So this particular stroma is responsible to the synthesis of glucose or the synthesis of starch. So means in a stroma what is the reaction is going to be taken place means carbon fixation or the carbon assimilation. So means here sir simply sir means, so in a stroma dark reaction takes place. Sir, dark reaction means which occurs in absence of light, sir means no dark reaction means here enna point na munnadi we see no light reaction. So means light reaction definitely takes place in the presence of light. Later ATP as well as NADPH will be synthesized, no. So by using of ATP as well as NADPH, so dark reaction starts, means that is not dependent on the light reaction but it is dependent on the products of light. அம்மா பார்த்து இப்போ இது கிரானமா, இது கிரானா, இங்கே கிரானாலே என்ன reaction takes place means light reaction takes place. light reaction sir means eppadi sir understand pannakanum means so light reaction means a light absorbed by these thylakoid membranes. Ipo thylakoids are there, no, each and every thylakoid ku or line irukku paatha. On the line, the line is nothing but membrane. On the membrane, now what we can find out means we can find out various types of pigments. After completion of this diagrammatical explanation, we will have that information here. Now on the membrane, different types of pigments are present. These pigments are capability, or these pigments has the capability to absorb the light. So in the presence of, or in the presence of light, grana receives the sunlight and starts to prepare the ATP as well as NADPH. So what are the ATP molecules as well as NADPH molecules are synthesized in the light reaction? All are comes to the stroma to start the process of dark reaction. So in grana light reaction takes place, and in in stroma dark reaction takes place. So dark reaction means, so you don't understand, it takes place in the presence of light. So why we are calling as a dark reaction means, so it is not depending on the light, but it is depending on the products of the light. Products of the light means, what are the products we are seeing? ATP as well as NADPH only, no. So thereby it depends on the ATP as well as NADPH to start the dark reaction, or simply to start the photosynthesis, or to start the preparation of food materials. Okay, so thereby here, first of all, in grana or on the grana, light reaction process takes place. So thereby what is happening? So thereby definitely ATP as well as NADPH which will be produced. Later dark reaction going to be started in the stroma. So one more time, so let us start to discuss. Sir, food materials are going to be synthesized means, sir, eppadi sir understand panagana? First of all, glucose will be formed. So that particular glucose later it will be converted into starch. Okay, so that particular information furtherly, so we will have a huge cycle at the time, let us discuss. But just part means where the process of photosynthesis occurs. So one kind of a cellaric null, already this one we have discussed in the cell unit of life, but we are discussing the process of photosynthesis, no, that's why kandipa we have to discuss this one right now. So now again come to the information. So come on to the information. Now let us see. Now here what is happening now? So there are different types of pigments are present. With the presence of pigments, so ATP as well as NADPH also are prepared in the grana. So that particular process only we are calling as light reactions or photochemical reactions. Photo means light. Photochemical reactions means the chemical reactions which takes place in the presence of light. No. So that's why those reactions we are calling as photochemical reactions. The latter are not directly light driven, but are the dependent on the products of light reactions ATP and NADPH. Later means, so here we are going to discuss under the stroma. So means here what is the point? The stroma is not directly light driven, means it is not dependent on the light, but are the dependent on the products of light reactions. Products of light reaction. Product of light reaction means products one is ATP and one more is NADPH. ATP as well as NADPH, these two are products of light reaction. So now here what is the point we are understanding now? Light reaction, it is dependent on the light. That's why we have given a name it as a light reaction. Or we have given a name it as a photochemical reactions. Photo means light. The chemical reactions means the chemical reactions which occurs in the presence of light. Now finally which one is synthesized? ATP as well as NADPH are synthesized. Now this particular ATP and NADPH2s are utilized in the stroma for the process of dark reaction. So that's why, hence to distinguish the latter means again dark reaction, they are known as the convention as dark reaction. So means because of that one, this particular entire reaction we are going to take it as dark reaction or carbon reactions. Carbon reactions. Now, however, this should not be construed to mean that they occur in darkness or that they are not light dependent. Means based on this, means by giving the name of dark reaction, we should not think like this. Means so these particular reactions are going to be taken place in a darkness, or these particular reactions are not dependent on the light reaction. Likewise, we should not take, even though name concerned, it is a dark reaction, but depending on the products of light. Directly it is not depending on the light, but definitely it is indirectly depending on the light. So that is a point to try to understand clearly. This particular diagram already we have discussed, but one more time, few more points or a few more labels are there now. Once let us discuss and let us leave them. Between the grana lines are there, no connection lines. Connection lines are there, no? Sir, what is the name of these connection lines means? We will call them as stroma lamellae. Stroma lamellae. Outer layer, our knowledge point of view, chloroplast double membranous cell organelle only, no? Rendu membranes irukku. Outer membrane and inner membrane. Okay, and between them gap is there, no? The gap here, the name we didn't write here, but we can be taken that gap as periplastidial space. Stroma, in a stroma dark reaction takes place, and in grana, no, on the grana light reaction takes place, already we have discussed. There, in chloroplast, no, we know very well 70s type of ribosomes are also present. Lipid droplets are there, fats or lipid droplets, and even starch granules also is there. Starch granule also is there. So likewise, it is one particular or the specialized cell organelle, so which is responsible to the process of photosynthesis. So photosynthetic pigments. Now let us start to discuss the photosynthetic pigments. So no worry, sir, this sir diagrams konjam customer don't think like that. Easy da. I'll explain in easy manner. Listen now. Now photosynthetic pigments, the separation of leaf pigments by any green plant through the chromatography or paper chromatography. So means here what we can do. Suppose if you would like to separate the, or if you would like to study the what are the pigments which are present in the leaf, if you would like to study, no, so definitely we can take the help of paper chromatography. So means paper chromatography is a technique, so to use or to separate the different types of pigments which are present in the leaf. This neat bit nalla save panniko. Means if you would like to separate the separation of pigments, if you would like to done means, so definitely what is the process we have to take the help? Paper chromatography process we need to use, or we need to start the process to know the pigments which are present in the leaf. Now the types of pigments concerned, so there are different types of pigments which we can be observed in the various types of leaves. Now let us learn roughly. First one is chlorophyll A. Actually neat information பார்த்தபோது கொஞ்சம் formula அப்புறம் what is the main important work that also discuss பண்ணிக்கலாம், but இது board information only, no, so that's why let us move so to learn the textbook information alone. So when we are going to discuss the neat information, so to discuss the about the pigments, so it takes 2 periods of time. Okay, so at the time let us concentrate, no worry. Now the pigments or pigments types point of view, chlorophyll A. So what is the color of chlorophyll A is bright or blue green color. Blue green or bright color. Next one, chlorophyll B, yellow green. Next one, xanthophylls color is yellow, and carotenoids color is yellow to yellow orange. இது எல்லாமே by heart பண்ணுக்கோ, எல்லாமே அண்டர்ஸ்டாண்டிங் பண்ண முடியாது, சரியா? ஸோ chlorophyll A color is bright or blue green, and chlorophyll B is yellow green, and xanthophylls color is yellow, and carotenoids color is yellow to yellow orange. Okay, Vama. Next one, pigments are அம்மா இது பை ஹார்ட் பண்ணிட்டீங்களா, லோன் பண்ணிக்கோ பை ஹார்ட்டோ ஒரு ஏதோ ஒன். So they will not ask us, but when we are asking questions to know at the time if you are responding we will feel happy, avladha. So what is the mode of learning you are following, that is your choice, but while asking questions no you must give answer, avladha. Next one, pigments or substances that have an ability to absorb light as at a specific wavelengths. So here definitely pigments, now only we will know there are different types of pigments, chlorophyll A, chlorophyll B, carotenoids, xanthophylls. Likewise we are learning various types of pigments which we can be learned or which we can be seen in leaf. Leaf. So likewise here these pigments or the substances have the capability to absorb sunlight and specific wavelengths. Sir, inge yellow wavelengths so these chlorophyll pigments or these pigments absorb light, sir, means no. What we are writing here, so definitely absorbs light, adi correct da, but not all wavelengths. So definitely at specific wavelengths. So nalla word nalla save panniko, only at a specific wavelengths only these pigments absorbs light, so very carefully. So now ipo paatha first side heading enna inge the absorbance, or simply you can take it as absorption of light by the chloroplast pigments, adi side heading ange enna information na. So we are going to discuss the graph related to the pigments, so how these pigments absorbs the wavelength. Now let us see. Now starting light for our knowledge, for our convenience to study, so we can be divided into 7 colors, VIBGR, we know very well. So ange wavelength approximately 400 to 770 nanometers, I think so, 390 to 770 nanometers, so we will study ange. So VIBGR appadina nearby the 700 nanometers blue color we will notice, and nearby of 700 nanometers we will see the red color wavelength. So that's why here what is the point, at a blue light and in a red light chlorophyll A and chlorophyll B absorbs more absorption of light or more light. So means here absorbs the more light, so where nearby the 400 nanometers and nearby the 700 nanometers. Sir, why sir, ange the main means ange, so we will notice the blue color and red color regions. Ipo VIBGR path VIBGYOR. So ange at a blue color region as well as in a red color region chlorophyll A and chlorophyll B absorbs more light. That's why here ipo paatha diagrams also label idu paathama. So even a graph also inge da adhigamaichi aprom dull aichi keela undi aprom again it has been increased. So means even chlorophyll A and chlorophyll B also appada da irukku. So idu nalla paathukka. Aprom based on this, did you remember one point? In earlier experiments we see T.W. Engelmann, he was used the prism and green alga cladophora, and based on the information what is point we have learned at a blue color region and in a red color region, so we will notice the more accumulation of green alga cladophora we see adi idida. Later, second one, rate of photosynthesis measured by the O2 release. O2 release and oxygen eppadi release so by the process of photosynthesis, that one showing in second diagram. Ipo second graph paatha, actually here 400 nanometers and here the 700 nanometers already we see in first graph. And madri ipo rate of photosynthesis paathambodhu where the more absorption of light will be there ange we will notice the more rate of photosynthesis. Ipo light kadiyadana eppadi photosynthesis process takes place, you only imagine. Ipo nalla adhikam light irukana, though absorption of light also will be there more, so thereby suppose if the light absorption is more means process of photosynthesis also will be more. Ipo process of photosynthesis is more means O2 means oxygen also will be released more. ஏமா இப்போ ப்ராசஸ் ஆஃப் ஃபோட்டோசிந்தசிஸ் ப்ராசஸ் டேக்கிங் பிளேஸ் மீன்ஸ் எப்படி ரொம்ப அதிகமாக லைட் அதிகமாக இருக்குன்னா லைட் அதிகமாக இருக்குன்னா கண்டிப்பாக ஆக்சிஜன் ஆல்சோ வில் பி ரிலீஸ்டு மோர் வை இஃப் மோர் ப்ரொடக்டிவிட்டி இஸ் தேர் ஆர் இஃப் மோர் ப்ராசஸ் ஆஃப் photosynthesis taking place means so while the process of photosynthesis are going to be occurred at the time oxygen will be released in more quantities ஏமா one by one link reactions மாதிரி ஏமா like I hope you understood that also. And third one, so third one, this one it indicates both, one is a rate of photosynthesis and one more is chlorophyll A involvement. இப்போ பார்த்தா at 400 nanometers the absorption wavelength is more by the chlorophyll A. Even in 700 nanometers also chlorophyll A absorbs more light. If more light is going to be absorbed at 2 wavelengths means in 400 nanometers as well as 700 nanometers. If inge blue light will be there and here the red light will be there. So in these two regions, so absorption of light will be more. So thereby in these two regions only we are noticing the more rate of photosynthesis. பார்த்தோமா இங்கே இப்போது இது ஃபஸ்ட் டயக்ராம்னா ஃபஸ்ட் கிராஃப் விச் இண்டிகேட்ஸ் chlorophyll A. அப்புறம்னா இங்கே in these regions 400 nanometers region and in 700 nanometers region light நல்லா அப்சார்ப் ஆயிடுச்சு. ஸோ தேர்பை இப்போ ரேட் ஆஃப் ஃபோட்டோசிந்தசிஸ் அங்கே தானே அதிகமாகும். ரிமைனிங் பிளேசஸ்னாலே பார்த்தா ரேட் ஆஃப் ஃபோட்டோசிந்தசிஸ் ரொம்ப டல்லாக இருக்குது. Remaining and in 400 nanometers region as well as in 700 nanometers region the rate of photosynthesis adhigam. So thereby here what is the point we can save in our mind means so definitely the red light as well as blue light or blue light as well as red light definitely has the more capability to absorb the light and definitely involves in the process of photosynthesis more or high rates. So that's why final point, what is the point we are learning from these diagrams? No, maximum absorption of chlorophyll A in the blue light and red light regions also shows high rate of photosynthesis or higher rate of photosynthesis. So this is a point here is a main. So based on these diagrams, so this is a point we can give finally. So thereby maximum absorption of chlorophyll A in the blue light as well as red light regions also shows the higher rate of photosynthesis. I hope you understood. So inge konjam 1 or 2 points inge kodukonu in the next video I will give you. So now this is information which we can learn under the pigments. What are the pigments playing an important role in the process of photosynthesis? Thank you.